Hello everyone. Today's topic is regulation of gene expression in eukaryotes. So general introduction is gene expression in eukaryotes is basically combination of three things, that is transcription of DNA to RNA, processing of transcribed mRNA, and translation of these mRNA to proteins. Now gene expression is basically the process in which the information encoded in the gene is used in synthesis of a gene product. Now this gene product can be a protein or an RNA. Then these regulation of these gene expression include a very wide range of mechanisms that are used in cells to increase or decrease the production of specific gene products. Now the process by which these information from gene is used uh, is is basically used in the synthesis of functional gene product. Now it can be classified based on their expression. These genes can be classified. It can be constitutive, meaning it can be called as housekeeping genes, and also controllable or inducible genes. Constitutive are basically they are expressed at a fixed rate irrespective of their cell conditions, and their structure is very simple. And then controllable or inducible is basically they are expressed when it is only needed, and their amount may decrease or increase with respect to their basal level in different conditions, and the structure is little complicated with some response elements. Now in terms of regulation, gene expression can be of two types basically: positive regulation, negative regulation. So talking about positive regulation, it is when the expression of genes is quantitatively increased by a presence of specific regulatory elements, which is known as positive regulation. And the elements that modulate these positive regulation, it can be called as a positive regulator or an activator. Now talking about negative regulation, it is when the expression of gene is diminished, or basically when it is suppressed in the presence of a regulatory element, that is called negative regulation. Now the element that mediates the negative regulation is called as a repressor. Now, why do we do this? The purpose of gene expression is adaptation and tissue specific differentiation and development. Now, adaptation is basically when this multicellular organism will respond to varying conditions. Such cells, when exposed to hormones and growth factors, they substantially change in shape, growth rate, and other characteristics. And to talk about tissue specific differentiation development is the genetic information present in each somatic cell of an organism is very identical. So the cells from these muscle or nerve tissues, they differ in their morphology and other properties very differently, but they contain their same DNA. So these diverse properties is the result of the difference in the gene expression. Now, expression of these genetic information is also regulated during ontogeny and differentiation of organism and its cellular components. Now, I would like to tell a few differences also of prokaryotes and eukaryotic gene regulation. Like the eukaryotes is a little more complex when compared to prokaryotes because of a lot of reasons, like it has a large and complex genome, it has uncoupled transcription and translation, then because of mRNA splicing, because of different cell types, because of chromatin structures which are more complex. So all these are few reasons why eukaryotes are more complex when compared to prokaryotes. Now moving on to the levels of the regulation of gene expression. Yeah. So levels of are as you can see, gene on DNA. These gene on the DNA will transcriptionally change to primary transcript, then changing into mRNA, which is highly active, which moves to the cytoplasm, translated, and it helps to and in the ribosomes, which is associated in the cytoplasm, and they are and here the mRNA will get translated to form polypeptides, proteins. So this is basically the whole level of regulation. in gene expression. Now we'll go detail one by one. So moving on, so in contrast to prokaryotic, gene regulation occurs primarily at a level of transcription. Initiation, regulation of gene expression can occur at many levels, okay, which can include alteration in the DNA template. It can include initiation of transcription. It can include mRNA transcription, modification and stability, synthesis and modifications, and stability of proteins which are formed. So all the primary event, the transcription of DNA to RNA occurs in the nucleus, and the RNA transcripts are modified by capping, polyadenylation, splicing. All these are post-transcriptional modifications, post-translation modifications. So here the resulting mRNA is then again, as I told, it's exported to the cytoplasm, where they become associated with these ribosomes. Ribosomes can be associated with the endoplasmic reticulum. Once it is associated with ribosomes, then mRNA are translated into the polypeptides, and the physical separation events of these gene expression makes it possible for the regulation to occur in very different places. Now moving on to first one, pre-transcriptional control. So talking about pre-transcriptional control, the first thing is chromatin remodeling. So chromatin remodeling, here what happens is basically these linear DNA, which are of 2 nanometer in size, they'll basically bind to histones. They'll bind to histones to form nucleosome. Then nucleosome will again tightly bind to form solenoids. Then solenoids will again bind to form scaffolds. Scaffolds will again condense itself to form chromosomes, the entire chromosomes. So basically here large regions of chromatin are transcriptionally inactive. Some cells are due to chromatin structure that hide DNA from transcriptional factors and enzymes. So when you're talking about chromatin, I would like to talk about two additional things. Basically the two types of chromatins, that is heterochromatin and euchromatin. Heterochromatin is highly compact and euchromatin is less compact. As I can say, heterochromatin is transcriptionally inactive. Euchromatin is transcriptionally active. Now, moving on to transcriptional control. Here is where enhancers and repressors come into play. So, here enhancers, they function by serving as binding site for regulatory protein. Enhancers is effective only in specific cell types which regulatory proteins are expressed. Enhancers can also exert positive influence on transcription even when they are separated by 1,000 base pairs from the promoters. Now, what are promoters? It is a region where the transcription initiation takes place. They are oriented either in the either direction, that is upstream 5' or downstream 3' end. Now, repressors are elements of regulatory protein that decrease or repress the expression of specific genes. These are identified as repressors. However, when these transcription factors bind to them, the expression of the gene when they control is reduced, or basically it is suppressed. So that's what repressors do. Now, as I told, these are the enhancers. These are the activators. So as I told before, these will enhance the activity of gene regulation. And in the case of repressors, when repressors are present, it is basically they will reduce, they'll just degrade or reduce its gene regulation. Now moving on to the post-transcriptional control. Here the initial RNA is not very functional. So the types of RNAs like these mRNA, tRNA, rRNA, which are formed, it will have to undergo a lot of modifications to become to become functional. So those modifications are phi capping, splicing, and poly A tailing. So phi capping means here a 7-methyl guanosine is added at the end of the mRNA, And splicing means it is basically removal of introns and bringing exons together. This will help to compress the structure of DNA. Why do we have to remove the introns? Is because introns are not expressed. So for a better gene regulation, we should remove, or splicing happens. Now, poly-A tailing is addition of polyadenine to 3 end of the mRNA. Um, and all these modification helps the DNA from degrading. Now, translation control, which is the last one. Here, mRNA is read and translated to proteins, and it takes place in a large ribonucleoprotein complexes called as ribosomes, which are present in the cytoplasm, as we all know. Then translation is divided into initiation, elongation and termination as normal. Now the main concept, or the central concept of these translation control, is that gene expression can be regulated by efficiency and also utilization of mRNA in specifying the protein synthesis. So that is why these main concept of translation is that. Now these are the references. Thank you.